Next topic, regulation of water balance. Regulation of water balance in the body is essential to maintain normal physiological functions. The body regulates water balance by adjusting the amount of water excreted through the kidneys, skin, and lungs. The regulation of water balance is achieved through a complex interplay between hormones and nerve system and the kidney. The main hormones involved in the regulation of water balance are antidiuretic hormone or ADH and aldosterone. ADH is produced by the hypothalamus and released by the pituitary gland in response to changes in blood volume or osmolarity. ADH acts on the kidneys to increase water reabsorption, thus reducing urine output and increasing blood volume. Aldosterone is produced by the adrenal glands in response to low blood volume or low blood pressure. Aldosterone acts on the kidneys to increase sodium reabsorption, which leads to increased water retention and increased blood volume. The nervous system also plays a role in regulation of water balance through the renin-angiotensin-aldosterone system or RAAS. The system is activated in response to low blood pressure and or low blood volume. The kidneys release renin, which converts angiotensin to angiotensin 1. Angiotensin converting enzyme or ACE then converts angiotensin 1 to angiotensin 2, which stimulates the release of aldosterone and causes vasoconstriction, increasing blood pressure and blood volume. The kidneys play a crucial role in the regulation of water balance by adjusting the amount of water excreted in urine. The kidneys filter blood to remove excess water and waste products and then reabsorb the necessary water and nutrients back into the bloodstream. The amount of water reabsorbed by the kidneys depends on the levels of ADH and aldosterone in the body, as well as other factors such as blood volume and blood pressure. In addition to hormones, the body also regulates water balance through thirst. And the body senses a decrease in blood volume or an increase in osmolarity, it triggers the sensation of thirst. Drinking water helps to restore the body's water balance by increasing blood volume and reducing blood osmolarity. So, in summary, the regulation of water balance in the body is a complex process involving hormones, the nervous system, and the kidneys. The body adjusts the amount of water excreted through the kidneys, skin, and lungs to maintain normal physiological functions. Hormones such as ADH and aldosterone play a crucial role in the regulation of water balance, and as do the kidneys and the sensation of thirst. Then, mechanism of concentration of urine. The concentration of urine is a critical process that occurs in the kidney to maintain water balance in the body. The mechanism of urine concentration involves the selective reabsorption of water from the nephron tubules in the kidney, which is regulated by a complex interplay of hormones and neural signals. This process is crucial in preventing dehydration as it allows the body to retain water by excreting concentrated urine. The mechanism of concentration of urine involves several parts, including the loop of Henle, the vasa recta, and the collecting duct. The loop of Henle is responsible for creating a concentration gradient in the medullary interstitium by reabsorbing sodium and chloride ions. The vasa recta is a network of blood vessels that surrounds the loop of Henle, which helps to maintain the concentration gradient by allowing for the exchange of solutes between the interstitium and the blood. The collecting duct is the final site of water reabsorption, which is regulated by the hormone vasopressin. The mechanism of concentration of urine begins with the filtration of blood in the glomerulus, where water and solutes are filtered into the Bowman's capsule. Then the filtrate then passes through the proximal tubule, where the majority of nutrients and ions are reabsorbed. The filtrate then enters the loop of Henle, which is divided into the descending and ascending limbs. In the descending limb, water is passively reabsorbed into the medullary interstitium, which is hypertonic due to the reabsorption of sodium and chloride ions by the ascending limb. As the filtrate enters the ascending limb, sodium and chloride ions are actively reabsorbed, which further increases the concentration gradient in the medullary interstitium. The vasa recta then maintains the concentration gradient by removing solutes from the interstitium and returning them to the blood. Finally, the filtrate enters the collecting duct, where the final regulation of water reabsorption occurs. This is regulated by the hormone vasopressin, which is produced in the hypothalamus and released by the posterior pituitary gland. Vasopressin acts on the collecting duct to increase the permeability of water, allowing it to be reabsorbed into the interstitium and ultimately excreted as concentrated urine. Then the regulation of water balance and the concentration of urine is a complex process that is controlled by both neural and hormonal mechanisms. The release of vasopressin is regulated by osmoreceptors in the hypothalamus, which detect changes in blood osmolarity. When blood osmolarity increases, vasopressin is released, which stimulates the reabsorption of water into the collecting duct. So, overall, the concentration of urine is a critical process that is essential for maintaining water balance in the body. It involves the selective reabsorption of water in the nephron tubules, which is regulated by a complex interplay of hormones and neural signals. Understanding this mechanism is crucial for understanding how the kidney functions to maintain homeostasis in the body. Next topic, countercurrent mechanism. The countercurrent mechanism is an essential mechanism for the concentration of urine in mammals. It involves the establishment of a concentration gradient within the renal tubule that allows for the reabsorption of water from the renal tubules and the formation of a concentrated urine. The countercurrent system involves both the loop of Henle and the vasa recta, which are specialized blood vessels that run parallel to each other. Parts involved. The loop of Henle is composed of a descending limb, a thin segment, and an ascending limb. The descending limb is permeable to water but not to solutes, while the ascending limb is permeable to water but actively transports solutes out of the water tubular lumen. So, descending limb is permeable to water but impermeable to solutes, while the ascending limb is impermeable to water and actively transports solutes out of the tubular lumen. The thin segment of the loop is highly permeable to water and solutes, allowing for the passive diffusion of these molecules across the tubular wall. The vasa recta is a specialized capillary network that surrounds the loop of Henle and serves to maintain the concentration gradient within the renal medulla. Then let's learn about the mechanism. The countercurrent system relies on the establishment of a concentration gradient within the renal medulla. The loop of Henle plays a crucial role in establishing this gradient as fluid flows down the descending limb of loop. Water diffuses out of the tubule and into the surrounding interstitial fluid, which has a high concentration of solutes. As the fluid flows up the ascending limb, solutes are actively transported out of the tubule, further concentrating the interstitial fluid. This establishes a concentration gradient that allows for the reabsorption of water from the collecting ducts in the medulla. The vasa recta serves to maintain this concentration gradient by providing a blood supply to the renal medulla without disrupting the concentration gradient. As blood flows through the vasa recta, it absorbs solutes from the surrounding interstitial fluid, further concentrating the blood. This helps to maintain the concentration gradient within the renal medulla, allowing for the continued reabsorption of water from the renal tubule. The countercurrent system begins in the descending limb of the loop of Henle, where water diffuses out of the tubule and into the surrounding interstitial fluid. As fluid flows up the ascending limb, solutes are actively transported out of the tubule, creating a concentration gradient within the renal medulla. The vasa recta then serves to maintain this concentration gradient by absorbing solutes from the surrounding interstitial fluid, further concentrating the blood and allowing for the continued reabsorption of water from the renal tubules. Then nerves and muscles associated. The countercurrent system is regulated by a variety of hormones and neural signals. Antidiuretic hormone or ADH is produced by the hypothalamus and acts on the collecting ducts in the renal medulla, increasing their permeability to water and allowing for greater water reabsorption. Aldosterone produced by the adrenal glands also play a role in countercurrent system by increasing the reabsorption of sodium ions from the tubule. The countercurrent system is also regulated by neural signals, with the sympathetic nervous system playing a role in regulating blood flow to the renal medulla. The countercurrent system is essential for the concentration of urine and the maintenance of water balance within the body. Without the system, the kidneys would not be able to produce a concentrated urine, leading to the loss of excess water and dehydration. The countercurrent system is also important for maintaining the osmotic balance within the body, as it allows for the reabsorption of both water and solutes from the renal tubules. Then countercurrent multiplier system. The countercurrent multiplier is a mechanism used by the kidneys to create a gradient of osmotic pressure which allows for the production of concentrated urine. It involves the interaction between two parallel portions of the nephron, the ascending and descending limbs of the loop of Henle. The countercurrent multiplier system involves the active transport of ions, particularly sodium chloride and potassium, in the ascending limb of loop of Henle. This creates a high concentration of ions in the medulla of the kidney, which draws water out of the descending limb of loop of Henle by osmosis. As the descending limb moves deeper into the medulla, the osmotic pressure gradient becomes increasingly steep, resulting in the further concentration of urine. The countercurrent multiplier system is made possible by the unique structure of the nephron, which includes a loop of Henle that dips down into the medulla of the kidney. The ascending limb of loop of Henle is thick and impermeable to water, while the descending limb is thin and permeable to water. The countercurrent multiplier system is regulated by the hormone antidiuretic hormone, which is produced by the hypothalamus and released by the pituitary gland. ADH increases the permeability of the collecting duct to water, allowing more water to be reabsorbed into the bloodstream and preventing the production of dilute urine. The countercurrent multiplier system is important because it allows the kidneys to conserve water by producing concentrated urine. This is particularly important in animals that live in arid environments and must conserve water to survive. So, in summary, the countercurrent multiplier system is a complex mechanism used by the kidneys to create a gradient of osmotic pressure which allows for the production of concentrated urine. It involves the interaction between the ascending and descending limbs of the loop of Henle, the active transport of ions, and the regulation of ADH. The system is important for water conservation and is essential for the survival of animals in arid environments. Then countercurrent exchanger. The countercurrent exchanger system is a mechanism that allows the efficient exchange of substances such as oxygen, carbon dioxide, and heat between two fluids that are flowing in opposite directions. The system is widely found in many biological systems, including gills of fish, the legs of birds, and the kidneys of mammals. The countercurrent exchanger in the kidney is the vasa recta, a network of small blood vessels that surround the loop of Henle in the renal medulla. The vasa recta is made up of two parallel capillary networks, one that descends alongside the descending limb of the loop of Henle and another that ascends alongside the ascending limb. The descending vasa recta is permeable to one that descends alongside the descending limb of the loop of Henle and another that ascends alongside the ascending limb. The descending vasa recta is permeable to water and solutes, but not to larger molecules such as proteins. As it descends into the renal medulla, it becomes progressively more concentrated due to the increasing osmotic gradient created by the loop of Henle. Water diffuses out of the descending limb of vasa recta and into the surrounding interstitial fluid, while solutes such as sodium and chloride diffuse into the vasa recta. The ascending vasa recta, on the other hand, is impermeable to water but permeable to solutes. As it ascends out of the renal medulla, it encounters a progressively lower osmotic gradient created by the loop of Henle. Solutes diffuse out of the ascending vasa recta and into the surrounding interstitial fluid, while water diffuses in. The countercurrent exchange system in vasa recta plays a crucial role in maintaining the concentration gradient of renal medulla, which is necessary for the production of concentrated urine. By allowing the efficient exchange of water and solutes between the vasa recta and the interstitial fluid, the countercurrent exchanger helps to maintain the osmotic gradient necessary for the reabsorption of water in the collecting ducts. So, in summary, the countercurrent exchanger in the kidneys, specifically in the vasa recta, is a crucial mechanism for maintaining the concentration gradient in the renal medulla, which is essential for the production of concentrated urine. By allowing the efficient exchange of water and solutes between the vasa recta and interstitial fluid, the countercurrent exchanger helps to maintain the osmotic gradient necessary for the reabsorption of water in the collecting ducts and ultimately the regulation of water balance in the body. Next, renal regulation of acid. Renal regulation of acid refers to the process by which the kidneys help maintain a proper acid-base balance in the body by regulating the concentration of hydrogen ions or H plus and bicarbonate ions HCO3 minus in the blood. The kidneys play an important role in regulating the pH of blood, which is a measure of its acidity or alkalinity. The normal pH range for blood is between 7.35 and 7.45, with a pH of 7.4 considered to be neutral. The kidneys regulate acid-base balance through three primary mechanisms: acid secretion, bicarbonate reabsorption, and the production of new bicarbonate ions. These mechanisms are carried out by various structures within the kidneys, including the glomerulus, proximal tubule, loop of Henle, distal tubule, and collecting duct. The primary mechanism by which the kidneys regulate acid-base balance is by excreting excess acid in the form of hydrogen ions and by reabsorbing bicarbonate ions or HCO3 minus. The proximal tubule of the nephron plays a major role in this process by actively secreting hydrogen ions into the lumen of the tubule and reabsorbing bicarbonate ions into the blood. The loop of Henle also plays a role in acid-base regulation by generating a concentration gradient that helps to concentrate urine and maintain a proper acid-base balance. The descending limb of the loop of Henle is permeable to water but not to solutes, while the ascending limb is impermeable to water but actively transports ions out of the tubule. This creates a concentration gradient that helps to maintain the proper balance of ions and pH in the body. The distal tubule and collecting duct are also involved in acid-base regulation, primarily through the actions of hormone aldosterone. Aldosterone helps to regulate the balance of potassium and sodium ions in the blood, which in turn affects the concentration of hydrogen ions and bicarbonate ions. So overall, the renal regulation of acid is an important process that helps to maintain the proper pH balance in the body. Dysfunction of the renal system can lead to a range of acid-base imbalances, including metabolic acidosis, respiratory alkalosis, and metabolic alkalosis. These conditions can have serious health consequences and may require medical interventions to correct. Then, renal regulation of base in detail. Renal regulation of base, also known as bicarbonate regulation, is a process that involves the kidneys in maintaining the acid-base balance of the body. The pH balance of the body is critical for proper physiological functioning, and any deviation from the normal pH range can cause various health problems. The kidneys play a vital role in regulating the body's acid-base balance by reabsorbing bicarbonate ions and excreting hydrogen ions through urine. The process of renal regulation of base involves several parts and mechanisms, including the proximal convoluted tubule, distal convoluted tubule, and collecting ducts and the hormone aldosterone. First, proximal convoluted tubule or PCT. The PCT is the first segment of the renal tubule where filtrate from the glomerulus enters. The PCT reabsorbs nearly 80% of filtered bicarbonate ions. In the PCT, bicarbonate ions are transported from the lumen to the interstitial fluid by the action of carbonic anhydrase, which converts bicarbonate ions and hydrogen ions to carbonic acid, which then dissociates into water and carbon dioxide. The carbon dioxide then diffuses into the cell where it is used to regenerate bicarbonate ions, which are then transported into the interstitial fluid. Then distal convoluted tubule or DCT. The DCT or distal convoluted tubule is a segment of the renal tubule that follows the loop of Henle. The DCT reabsorbs the remaining 20% of bicarbonate ions primarily through the action of the Na plus or H plus antiporter. Hydrogen ions are exchanged for sodium ions, which are then transported out of the cell and into the interstitial fluid. The hydrogen ions combine with the bicarbonate ions in the interstitial fluid to form carbonic acid, which dissociates into carbon dioxide and water. The carbon dioxide then diffuses back into the cell where it is used to regenerate bicarbonate ions. Then collecting ducts. The collecting ducts are the final segment of the renal tubule where the urine becomes concentrated. The collecting ducts reabsorb any remaining bicarbonate ion and excrete excess hydrogen ions. The amount of bicarbonate ions reabsorbed and the amount of hydrogen ions excreted depend on the body's acid-base balance. Then aldosterone. Aldosterone is a hormone produced by the adrenal glands that regulate the reabsorption of sodium and the excretion of potassium in the kidneys. Aldosterone also indirectly affects the reabsorption of bicarbonate ions in the proximal tubule and the distal tubule. Aldosterone promotes the reabsorption of sodium, which indirectly increases the reabsorption of bicarbonate ions. The renal regulation of base is important in maintaining the body's acid-base balance and preventing acidosis or alkalosis. Acidosis is a condition that occurs when the pH of the blood drops below the normal range, while alkalosis is a condition that occurs when the pH of the blood increases above the normal range. Both conditions can lead to serious health problems, and the renal regulation of base helps prevent these conditions from occurring. So, in summary, the renal regulation of base involves the reabsorption of bicarbonate ions and the excretion of hydrogen ions to maintain the body's acid-base balance. The process involves several parts and mechanisms, including the proximal convoluted tubule, distal convoluted tubule, collecting ducts, and the hormone aldosterone. The kidneys play a critical role in regulating the body's acid-base balance, and any malfunction can lead to severe health problems. Then renal regulation of acid and base balance. The renal system plays an important role in the regulation of acid-base balance in the body. It maintains the pH of blood within a narrow range of 7.35 to 7.45 by removing excess acid or base from the body. The regulation of acid-base balance is achieved by three main processes: buffering, respiratory regulation, and renal regulation. Buffering is the first line of defense against changes in pH. Buffers are substances that can either absorb excess acid or base to prevent a change in pH. The most important buffer in the body is bicarbonate or HCO3-, which is present in both the blood and the renal tubular fluid. Bicarbonate combined with excess acid to form carbonic acid H2CO3, which can be further broken down into water and carbon dioxide. This reaction can be reversed to release bicarbonate ions to neutralize excess base. Then respiratory regulation involves the regulation of carbon dioxide levels in the body. Carbon dioxide is an acid and can combine with water to form carbonic acid, which can dissociate into bicarbonate and hydrogen ions. If the body produces excess acid, the respiratory system can increase the rate and depth of breathing to remove excess carbon dioxide and decrease the acidity of the body. Then renal regulation of acid-base balance involves the reabsorption and secretion of ions in the nephron of the kidney. The renal tubular cells can secrete hydrogen ions RH plus into the urine and reabsorb bicarbonate ions HCO3- back into the blood to maintain the acid-base balance. This process is regulated by several mechanisms, including the reabsorption of bicarbonate ions in the proximal tubule and the secretion of hydrogen ions in the distal tubule. In the proximal tubule, bicarbonate ions are reabsorbed through the sodium bicarbonate co-transported. Sodium ions and bicarbonate ions are transported together into the epithelial cells and then into the blood. This process creates a bicarbonate gradient that favors the reabsorption of bicarbonate ions from the tubular fluid. From the distal tubule and collecting duct, hydrogen ions are secreted into the tubular fluid through the action of the H plus ATPase pump. This pump can transport hydrogen ions against a concentration gradient, creating a high concentration of hydrogen ions in the tubular fluid. The reabsorption of bicarbonate ions and secretion of hydrogen ions in the nephron is regulated by hormones such as aldosterone and antidiuretic hormone or ADH. Aldosterone is a hormone secreted by the adrenal gland in response to low blood pressure. It increases the reabsorption of sodium ions in the distal tubule and collecting duct, which creates a concentration gradient that favors the secretion of potassium and hydrogen ions into the tubular fluid. This process increases the excretion of acid and helps to maintain acid-base balance. ADH is a hormone secreted by the pituitary gland in response to changes in blood osmolarity. It increases the water permeability of the collecting duct, which allows for the reabsorption of water and concentration of urine. This process can also increase the concentration of ions in the tubular fluid, which can affect acid-base balance. So, in conclusion, the renal system plays an important role in the regulation of acid base balance in the body. The reabsorption and secretion of ions in the nephron can help to maintain the pH of the blood within a narrow range. The regulation of acid base balance is a complex process that involves multiple mechanisms, including buffering, respiratory regulation, and renal regulation. The understanding of these mechanisms is important in the diagnosis and treatment of acid base disorders. Then, renal electrolytic balance. Renal electrolyte balance refers to the regulation of the concentration of various electrolytes in the body fluids and through the filtration and reabsorption process in the kidney. The electrolytes include sodium, potassium chloride, calcium, magnesium, bicarbonate, and phosphate. The balance of these electrolytes is essential for the normal functioning of various physiological processes in the body, such as nerve and muscle function, pH balance, and fluid balance. This essay will discuss the parts involved in the renal electrolyte balance, the mechanism, procedure, and the importance and maintaining parts involved in renal electrolyte balance. The renal electrolyte balance involves several parts of the kidney, including the nephron, juxtaglomerular apparatus, and collecting duct. The nephron is the functional unit of kidney that filters the blood and produces urine. It is composed of the glomerulus, Bowman's capsule, proximal tubule, loop-of-Henle, distal tubule, and collecting duct. The juxtaglomerular apparatus is located at the junction of the afferent and efferent arterioles and of the glomerulus, and it plays a crucial role in regulating blood pressure and electrolyte balance. The collecting duct is responsible for the final concentration of urine and regulation of electrolyte balance. The regulation of electrolyte balance in the kidney involves several processes, including filtration, reabsorption, and secretion. Filtration occurs in the glomerulus, where blood is filtered through the capillaries into the Bowman's capsule. The filtrate contains various electrolytes that are then reabsorbed back into the bloodstream through the tubules of the nephron. The reabsorption process occurs primarily in the proximal tubule, loop of Henle, and the distal tubule, where different electrolytes are reabsorbed using specific transport mechanisms. The secretion process occurs in the distal tubule and collecting duct, whereas excess electrolytes are secreted into the urine to maintain the electrolyte balance. Then, procedure. The regulation of electrolyte balance in the kidney is influenced by various factors such as hormone secretion, the concentration of electrolytes in the body fluids, and the volume of fluid intake. The hormone aldosterone, for example, is secreted by the adrenal gland in response to low blood pressure and low sodium levels. It increases the reabsorption of sodium and the secretion of potassium in the distal tubule and collecting duct. Antidiuretic hormone, or ADH, is another hormone that regulates electrolyte balance by increasing the reabsorption of water in the collecting ducts. The regulation of electrolyte balance in the kidney is essential for maintaining normal physiological processes in the body. Electrolytes such as sodium and potassium play a crucial role in maintaining nerve and muscle function. Calcium and phosphate are necessary for bone health and other physiological processes such as blood clotting. The balance of this electrolyte is also essential for maintaining the pH balance of the body fluids. Electrolyte imbalances can result in various health problems such as muscle weakness, seizures, cardiac arrhythmias, and bone disorders. So, in conclusion, renal electrolyte balance is essential for maintaining normal physiological processes in the body. The process involves several parts of the kidney, including the nephron, juxtaglomerular apparatus, and collecting duct, and is regulated by various factors such as hormone secretion, the concentration of electrolytes in the body fluids, and the volume of fluid intake. The maintenance of electrolyte balance is crucial for the maintaining and functioning of various physiological processes in the body, and any imbalance can result in various health problems. Then, structure of urinary bladder. The urinary bladder is a hollow muscular organ situated in the pelvic cavity of human body. It plays a vital role in the storage and expulsion of urine from the body. The structure of the urinary bladder is composed of several layers of tissues that work together to carry out its function efficiently. The urinary bladder is a muscular sac that is distensible and can hold varying volumes of urine depending on its capacity. The shape of the bladder can be described as a pear-shaped or ovoid structure that is wider at the base and narrow at the apex. The base of the bladder is attached to the pelvic floor muscles, while the apex is directed towards the pubic bone. bladder wall is composed of 4 layers of tissue, namely the mucosa, submucosa, muscularis, and serosa. The innermost layer or mucosa is lined with transitional epithelium, which is a specialized type of tissue that can stretch and contract in response to volume of urine. The mucosa layer also contains glands that secrete mucus to protect the epithelium from the acidic urine. The submucosa layer lies beneath the mucosa and contains blood vessels, lymphatics, and nerves. The muscularis layer is composed of smooth muscle fibers that are arranged in 3 layers, namely the inner longitudinal, middle circular, and outer longitudinal layers. These muscle fibers contract to expel urine from the bladder during micturition. So, the serosa is the outermost layer of the bladder wall and is composed of connective tissue that covers the muscular layer. The bladder is supplied with blood by the internal iliac artery and is innervated by the parasympathetic and sympathetic nervous systems. The parasympathetic nerves from the sacral spinal cord stimulate the constant contraction of the bladder wall and relaxation of the renal internal urethral sphincter, leading to the release of urine from the bladder. The sympathetic nerves from the thoracolumbar spinal cord thoracolumbar spinal cord control the relaxation of the bladder wall and contraction of the internal urethral sphincter, thereby preventing the release of urine. The bladder also contains 2 openings, the ureters and the urethra. The ureters are muscular tubes that connect the kidneys to the bladder and transport urine from the kidneys to the bladder. The urethra is a muscular tube that connects the bladder to the external urethral orifice and is responsible for the elimination of urine from the body. The urethra is shorter in females than in males, which makes females more susceptible to urinary tract infections. So, in conclusion, the structure of the urinary bladder is composed of several layers of tissues that work together to store and expel urine from the body. It is a vital organ that plays an important role in maintaining the fluid balance of the body. The bladder is innervated by parasympathetic and sympathetic nerve systems and is supplied with blood by the internal iliac artery. Understanding the structure of the urinary bladder is crucial in the diagnosis and treatment of urinary tract disorders. Next topic we will be learning with the help of next video. Thank you for listening.